Well, I'm going to talk about something very different, and you'll see that I've got a question mark on my title slide, because a lot of physicists feel that there is no place for biology in physics. And the take-home message today really is that you should see things otherwise, and if I succeed in doing that, I'll go home happy. Now, I've actually got a lot of history on my side in claiming that physicists ought to take an interest in biology, because what I've done here is to make a list of the people, really the people who developed the physics curriculum as it is today. And in red, I've put a note of the contributions they made in physiology and medicine. So people tend to forget, for example, that Galileo started life as a medic, and all of his interest in pendulums and that sort of thing was stimulated by the fact that he wanted to time the arterial pulse. Now, it hasn't always been an enormously successful interaction, I'm forced to admit, because Descartes came up with a theory of nerve conduction which involved the movement of some mysterious fluid. And if he ever stopped to inspect his McDonald's chicken leg, he'd have realised that he was barking up the wrong tree there. But you see, people like Young worked both on blood flow and notably on optics. And Young actually almost blinded himself in his efforts to understand how the eye worked. And he actually left instructions to his assistant to remove his eye and do a crucial experiment when he died. So it's the ultimate experiment. You never have to know that you're wrong. And then Helmholtz, you see, not only was the father of many areas of physics, but he also developed a theory of hearing, which is very much what we teach today. And he was at various times a professor both of physics and of physiology. And I hope in the near future we may find more and more people who work in both fields. Well, the big challenge in biophysics these days is to understand the physics that's going on inside the cell. I'm sure you've seen this sort of diagram. And over the last few years, the cell and molecular biologists have given us a great deal of information about all the clever chemistry that's going on in all of the internal compartments of the cell. But if you think about it, all they've left us with is a library of what the cell can do. It doesn't tell us either how the cell actually does things or how it decides what to do next. And those are questions that need physicists to understand. Now, the clever chemistry that cells do actually involves working with countably small numbers of molecules and working with reactions very, very close to the thermal noise level. So the cell only works because it can get these few molecules in the right place at the right time. And we need physics to understand that. The big problem is that it's going to need new physics as well, because statistical mechanics, even quantum mechanics, is not really up to answering some of these biological questions. Now, I'm going to leave that as a challenge for you to do, and I'm going to concentrate today on the applications of a very elementary bit of physics, the way that you can apply mechanics to understanding biological systems. And to find interesting mechanical problems, you don't have to look any further than the membrane that defines the envelope of the cell and all the internal compartments. And this, as Eros already showed, is a structure just 2 molecules thick. But they're remarkable molecules. They're these things called phospholipids, which are molecules with a hydrophilic head and a hydrophobic tail. So the head wants to be in contact with water, and the tail desperately wants to avoid contact with water. Now, until the 1970s, the chemists didn't take these hydrophobic interactions very seriously. It was a very weak interaction. So they thought it couldn't possibly be doing anything interesting. That may be true on the level of the individual molecule, but when you sum the contributions of vast numbers of molecules in a cell membrane, it becomes a dominant force. And it's actually sufficient to ensure that cell membranes form these bilayer structures spontaneously. So there you have one of the first steps in the evolution of life. Now, it also gives this membrane very interesting mechanical properties. It behaves as a 2-dimensional fluid. You might like to go home and think tonight what mechanical properties you'd expect of a 2-dimensional fluid. Whether or not you succeed, let me just point out how important these are to the survival of cells. Red cells, the very simplest cells in our body, you always draw as these biconcave discs. That's absolutely true when they sit on bottom of a test tube. But every time they pass through capillaries, the capillaries are as small as the cells, so these cells get stretched completely out of shape. And over its lifetime, a red cell has got to undergo this sort of deformation about 1 million times. So in purely engineering terms, this is a major mechanical feat. Now what we're very interested in Exeter is relating the mechanical properties of the cell membrane to its detailed structure. And if you've played around in the second year labs, measuring Young's modulus on pieces of wire and that sort of thing, you might think it's a tall order to measure an elastic modulus on a structure just 2 molecules thick. There are actually a number of very elegant ways of doing just that. And I'm going to be selfish and just talk about a method that we've developed in Exeter. And this is something that we call, when my slide works, fluctuation spectroscopy. What you've got in the top left-hand corner, I have an 8. This is a red cell sitting on the stage of a microscope. And this is supposed to be a snapshot from a video, which is obviously not running. Were it running, you would see that the cell membrane is so soft that the thermal motion in the fluid around it sets the membrane into constant vibration. And we can analyze the modes of vibration of this membrane. If you like, we can do a Fourier analysis of the various modes that this membrane can take on. And we can put those modes together with a mechanical model of the cell membrane. This models the cell as a network of springs with hinges between them. And we can relate the fluctuation spectrum to the various moduli you need to characterize the membrane, its behavior and bending and shear and that sort of thing. Now, when we first did this, we thought a very nice bit of physics, and we were very impressed with the results. But we're now working with people at the hospital to see whether or not changes in mechanical properties of the membrane in diseases like diabetes, oxidative stress, and so on, can be detected by this sort of technique. So this sort of measurement can provide a marker of disease and its progression. And that's one of the great justifications for working in this field. You don't have to continually justify what you're looking at. Now, the cell membrane has to do more than just withstand mechanical forces. Cells use mechanical forces as signals, tell them where they are and what they've got to do. And this is brought about by the proteins in the cell membrane. The lipid bilayer itself does a very good job of separating the inside and the outside of the cell. So when cells actually want to exchange molecules, they use channels formed by transmembrane proteins. And what's particularly interesting to the biophysicists is that there are a family of these channels which are opened by electrical and mechanical signals. So when you pull on the membrane, you open the channel, and that's a signal that the cell is experiencing some sort of mechanical stress. Now these signals are used in normal processes like tissue development, bone growth and that sort of thing. They're used in hairing. Blood vessels spend an awful long time measuring the flow properties over the surface using this sort of channel. But also things go wrong with these channels in diseases, in things like cardiovascular disease and cancer. So there's an awful lot of interest at the moment in trying to understand how these channels work. And this is a cartoon taken from a Nature paper of a few years ago, suggesting various mechanisms. And you can see there's a lot of fun for physicists in trying to distinguish between these various mechanisms. The only point I'd make tonight is that a lot of these mechanisms involve coupling the membrane to things either on the inside of the cell or on the outside. And that gives me an excuse to talk about another major area of work in Exeter, which is on the extracellular matrix. Now, 90% of the world don't know the extracellular matrix exists. But every tissue of the body is made up of cells and a bit that's not cells called the extracellular matrix. And in things like muscle, the extracellular matrix is just the glue between cells, holding them together and giving them something to pull on. But if you look at tissues like cartilage, the matrix is about 90% of the volume and the cells are just 10%. So they're not really terribly important. And this is the structure of tissues seen in a high-quality electron microscope. Here you've got a cell embedded in its extracellular matrix, which consists of a network of fibrous proteins embedded in a viscoelastic gel. And there are two sorts of fibrous proteins. Very stiff fibers of a material called collagen, which is the gristle in your McDonald's chicken, and elastin, which is a very elastic protein. And this extracellular matrix does an awful lot of interesting physics. It determines the mechanical properties of tissues, which are vital in things like cartilage, and it also regulates the ways that nutrients and other molecules are exchanged between cells and their environment. And so we're working on a lot of aspects of these physical properties of the extracellular matrix, but I said today I would talk about mechanics. And biomechanics is a subject that evolved in the 1960s. It actually evolved when the American aerospace industry went belly up, and a lot of engineers found themselves with expensive mechanical testing equipment and a lot of time on their hands. And so they started characterizing the mechanical properties of things like blood vessels, tendons, and cartilage and things. They didn't actually make a lot of sense of what they were measuring, but they did, very importantly, establish that changes in the mechanical properties of tissues are associated with vascular diseases, arthritis, and many other things. Now, since the 1960s, the biochemists and cell biologists have accumulated vast amounts of information about what the extracellular matrix is really made up of. But again, they need physicists to come along and tell them what it's all doing. So the current challenge is to understand the mechanical properties of tissues on a microscopic level, or at the level of individual molecules, and to see how cells are coupled into this system. And that's something that's now become technically possible, not least because of new forms of microscopy, some of which we've pioneered here at Exeter. We're great enthusiasts for the techniques of nonlinear microscopy. Very crudely, and I'm undoubtedly offending Bill sorely, nonlinear microscopy is all about getting multiple photons to interact with your target molecule, so that the light which is scattered from the target molecule provides you with a fingerprint of the chemistry of that molecule. I'm very happy to talk about the details of these methodologies later on, or to show you around the microscope systems we use in Exeter. What I've just got time for at the moment is to show you why this is so important.
No offense, Peter, it's an excellent.
Anyway, this is a wonderful way of getting pretty pictures. Cartilage, small blood vessels, even fat tissue shows up beautifully. But there's much more to it than this, because we're not using stains and things. These tissues are all alive while we're looking. So we can do this mechanical testing on the stage of our microscope and see what actually moves when we apply mechanical loads, so we can begin to work out the functions of all the molecules in the tissues. Now, we're funded by the ARC to look at changes that go on in cartilage in the development of osteoarthritis, which is a disease that's probably going to catch up with a lot of you in the future. We're funded by the British Heart Foundation, looking at small blood vessels, telling them that we're going to learn something about the changes that occur in diabetes and heart disease, which again are diseases that are going to catch up with an awful lot of you. And we're working on adipose tissue, which is of interest to those of you who are just going to get fat. Because there's a very fashionable theory at the moment that mechanical properties of fat tissue are all to do with where and how much you store the dietary fats. So there are very interesting reasons for looking at the mechanical properties of all these tissues. And this is very much work in progress, and I'm happy to talk to you about more anytime you care to drop in and take a cup of tea with me. But I'd like to finally move on to another aspect of our work. I've tried to make the point that biological molecules do very clever things. And one thing that they do very cleverly is to make long-range elastic materials. Long-range elasticity is obviously important to the skin, to the blood vessels, to the lungs. And if you work out how many times your lungs are going to expand and deflate in your lifetime, that's again a stiff mechanical challenge. And nature uses this protein I already mentioned, called elastin, to serve all these functions. And it's a better example of rubber than rubber is. So we need to understand what's special about it. Well, you probably realise that you can make elastic materials either by using energy to drive the elastic recoil, which is the way that metals and such like work. Very good for short-range elasticity. It's not very good for long-range elasticity. So rubbers use entropy elasticity. As you stretch a rubber, you produce a more ordered structure. You reduce the number of conformational degrees of freedom so that entropy can drive the elastic recoil. Well, there's some very simple thermodynamics you can do to prove that this goes on in elastin. It's a sample of entropy elasticity, but it's cleverer than rubber because elastin also contains a lot of hydrophobic amino acids. And we've already seen how important hydrophobicity is in biology. And these molecules form little sort of oil droplets along the molecule, so that as you stretch the molecule, you're forcing these hydrophobic domains, instead of being nicely tucked up and secure away from water, into contact with water, so they can't wait to contract. So this hydrophobic connection also drives contraction. Now, elastin is not this sort of random network. This is a micrograph of an elastin fibre, and you see it's a highly ordered structure. So what we need to know now is exactly what's going on at the molecular level inside this sort of fibre. And the way we can approach that sort of problem is to use spectroscopy, which you've already heard about tonight. Bill mentioned Raman Spectroscopy, which is this vibrational spectroscopy. It's inelastic scattering of light. Raman got a Nobel Prize for this, by the way, so it's worth understanding. You set the target molecule in vibration, and the spectrum of inelastically scattered light tells you something about the vibrational structure, that is the bonds within the molecule. So this is a Raman spectrum, which provides a chemical fingerprint of your molecule. Now, with modern technology, as Bill pointed out, you can acquire spectra from planets. We look the other way and acquire our spectra through a microscope. So we can do these sort of mechanical testing experiments. We can stretch fibres on the stage of a microscope and see how these spectra change. And that tells us a lot about what is happening to the molecules. Some of these peaks tell us about the structure of the molecule, so we can see if we're just uncoiling the molecules. Turns out that doesn't happen. We can see the spectrum is polarized, so we can look at polarization changes, and we can see little side chains on the molecules do move about as we stretch the molecules. But it turns out that this little bump here is a peak due to water interacting with the molecule. And that's the thing that really changes as you stretch the molecule. So we can now see at a molecular level what's going on. Now, you see this spectrum stops rather abruptly at the low frequency side. That's because of technical limitations in the bandpass characteristics of the filters in our microscope. But it's a bit frustrating because these very low frequency modes tell you about the ways that the whole molecule can vibrate. They tell you about whole molecule vibrational properties. So we're working now just to get better spectrometers so we can get round to this region. But there's another way you can look at these whole molecule vibrations. And since the appointment of a new lecturer in my group, we're the first lab in the world, actually, to use this technique of Brillouin scattering to look at the mechanical properties of some biological tissues. Brillouin scattering, I'm sure you all know, is another form of inelastic scattering. And it's inelastic scattering because you're propagating sound waves through your material. And you also remember that the speed of sound provides you with a measure of the mechanical properties of the material the sound waves are traveling through. And so we can determine the sound speed from the spectrum of inelastically scattered photons and use that to characterize the mechanical properties of our material. We can actually determine, using this sort of geometry, which I won't go through in detail, all the mechanical components of the elastic tensor. So this provides us with a complete characterization the mechanical properties of biological tissue on a microscopic scale. The big problem is we've started to do this. We don't have the faintest idea what our measurements mean, because we're now exploring mechanics on a length scale and a frequency scale. This is all working at gigahertz frequencies that we've never used before. So we've got to try and interpret those observations. And that may well be a challenge that I'll have to hand over to people of your generation. But nevertheless, we feel that this sort of measurement is likely to have some sort of diagnostic application. So just to finish up, I've tried to tell you that there are applications of physics in biology at many levels. We can understand mechanics at the whole tissue level, which is important for understanding diseases, important for improving the design of prostheses. We can understand mechanics at the level of the individual cell, and the sort of holy grail of this sort of research is that we can mechanically talk to cells so that we can subvert various disease processes, but that's very definitely a challenge for you. And we can also look at mechanics at the level of individual molecules. We can learn about the dynamics of proteins, what they do clever, and see whether or not we can take some of these lessons to design novel polymers for biological, biomedical, or other purposes. Well, that's a to-do list for you all, so I think I'll shut up. Okay, well, thank you very much.